All right, everybody, welcome to our July 2018 micro seminar, July 25th, 2018 micro seminar. Today we're going to be hearing from Ben Temperton. Before I get to Ben, I just want to point out that we have a full slate of micro seminars coming up for the rest of the year, and actually I think into next year. So take a look at the microseminar.wordpress.com page to get a sense of what's coming up. We have on the docket Rebecca Case, Mario Tubez Rodrigo, Jen Glass, Luis Zaman, and Kat Milligan-Meyer, all in the future. And as always, if you're interested in presenting, or you have a student that you would like to present, or a postdoc, or any kind of a trainee, please get in touch with us, either myself or David Baltris or Lizzie Wilbanks. We'd love to hear from you. Let's get back to today's seminar. It's going to be entitled Sequencing the Smallest Elephant in the Room. Our speaker today is Ben Temperton, a lecturer. For those of you who don't know, that's the equivalent position of a assistant professor at Exeter University in the UK. Ben did his PhD at, he was based out of the Plymouth Marine Labs, but technically his degree came from Belfast. And he was co-advised by Jack Gilbert and John McGraw. He did a master's before that at Nottingham University, and that was focused on IT. So he actually has a lot of experience in programming from a strict computer traditionalist perspective, I guess. And he did a chemistry degree at the University of Birmingham. So Ben's been all over the place, and he brings a wealth of different experience to the task. And so without any further ado, I will turn it over to you, Ben. Take it away.
Great. Thanks, Cameron. And thanks for the invite for coming to do this. It's super exciting to be part of the micro seminar series. I've been following it for a very long time. And yeah, it's kind of nice to be giving to it, having followed a few of them. So, right, I'm just going to go see if I can share my screen now. And do that. So, are we good? Can we see that?
Perfect. Yeah, just hide that.
Okay.
There you go. Go for it.
Thank you. So the reason I've called my talk "Sequencing the Smallest Elephant in the Room" will hopefully become apparent, and it's really focusing on the viruses that infect the microbial, the bacterial communities. That this is going to predominantly focus on the marine environment, but the smallest elephant in the room, I put it there because essentially I think the viruses are a largely forgotten component of the microbial communities. And part of that is because of the difficulty in analyzing some of these things. So what I've been doing since I became a lecturer at Exeter is trying to find better ways to capture viral metagenomics in order to try and improve how we resolve these kind of communities, and then try and make it a bit easier so people can start incorporating them into their microbiome work. So why is this important? Well, let's start by saying there are estimated to be 10 to the power 31 viruses on Earth. Now, there's an absolutely enormous number that the human brain rather struggles to get round. So, so let's try and find a way of kind of getting that number into people understand it. And this here is and it's called the Virgo supercluster. And this galactic group here is where our galaxy is, and so is the Andromeda galaxy. It's about 54 galaxies make up this local galactic group. And you can see these other galaxies within this within the Virgo supercluster. It's 100 million light years, so it's a big space. But if we imagine now that you have a 10 to the 31 viral particles on Earth, and you roughly assume everyone is about 200 nanometers in length, and then you stack all those together, and then divide by the numbers of meters in a light year, you actually work out that you have 200 million light years worth of viruses spanning this. So if you put all the viruses together, you will happily go across this, across the Virgo supercluster and back almost to the beginning again, just on the viruses you've stacked. So even though they're tiny, there's huge numbers of them about. And one thing you will learn when you listen to virologists talk is they love to extrapolate to enormous numbers from rather small sample sizes. So there's probably an order of magnitude either way on this, that we're over or underestimating viral abundance. But whichever way you slice it, there's a lot of viruses out there. Okay, so in seawater, where most of the work has been done, in the surface, there's around 10 to the 7 viruses per mil. That decreases with depth due to the reduction in the cells that they prey upon. And then in the human gut, it's about 10 to the 9 viruses per gram of feces. So again, the human gut is hugely abundant for viral particles, and they have a very important role we're finding out now in terms of the virome changes with disease states and so on and so forth. But I'm not going to be concentrating on those in this particular talk. And then in soils, again, 10 to the 9 viruses per gram of soil. So you imagine how many grams of soil there are on Earth. There's an awful lot of viral particles out there. And the vast majority of these viruses are those that infect bacteria. So we call those bacteriophage. This is a picture here of a virus you may be familiar with called the T4 virus. T4 myoviridae-like virus is infecting a particular cell. And they attach the cell and inject their DNA. And then once that DNA is in that cell, it then convinces that or takes over the metabolism of that cell to create new viral particles, breaks the cell open, and then continues to propagate the viruses that way. Okay, so I guess we're living in an age now where the microbiome is becoming a major important area of research for both human health and agriculture and food security and so on and so forth. And I think this is there's a great book by Ed Yong called I Contain Multitudes that really focuses on the importance of microbes in maintaining some of these systems, particularly in human health. And there's also a lot of work done by Jack Gilbert and Rob Knight's group looking at the Earth Microbiome Project. You may have seen some of these where they're swabbing the inside of the mouths of Komodo dragons to see what grows there to try and capture the viral diversity on Earth. And we've seen a lot of it now done in marine systems, because largely concentrating cells from marine systems is very easy. You just simply filter 20 liters of seawater and then extract the DNA from it. And then we're starting to get into soils where we find huge diversity of microbial groups in there. Now, the one thing that seems to be missing from almost all of these studies is the viruses. It's very rare to find a paper, certainly in the medical world and even in soils and that actually starts talking about viral diversity in these systems and actually what the role of the viruses are in terms of the ecology of maintaining predator-prey interactions and so on and so forth. So, in my opinion, as any zebra will tell you, it's a very bad idea to ignore the predators in a system if you want to understand how it works. And viruses are certainly no different in this regard. So they undergo, or they participate in classic Locke-Volterra type dynamics, whereby your cellular abundance will increase, and then your virus abundance will increase to predate on them. That will then drop the abundance of your prey, and then the viruses decrease. And so on and so forth. In what we call the kill the winner model, where actually the viruses predate on the most abundant bacteria in that system. And it's all to do with the fact that essentially, in order for a virus to infect a cell, it has to basically connect with that cell at some point. So there's a density-dependent interaction there. Now, what this does is it actually, by knocking out the winner, it increases the diversity of that system. So viral predation actually increases the amount of different types of microbes that you get in a system. And that's the same with foxes and rabbits and lions and zebras and everything else. But viruses also have some pretty cool tricks up their sleeve. And this is from an excellent paper by Maya, where she came up with some cool cartoons that kind of show this work. And so we know that the viruses are lysing open the bacteria and essentially releasing all the stuff that was inside that cell. And we estimate it's around 10 billion tons of carbon per day get released from marine bacteria doing this. So you imagine that's incredibly important for global carbon biogeochemistry. The viruses also have some tricks that would make lions pretty jealous. One of them is actually when they infect the cell, they have something on them called auxiliary metabolic genes. And these genes that actually enable them to take over their host metabolism and then essentially improve the capacity of that cell to generate new viral particles. So a classic example might be a virus that encodes a phosphate transport system. So the virus goes into the cell, starts upregulating phosphate transport. That cell then brings phosphate into the cell, and that allows for the creation of more viral particles because viral particles tend to be phosphate rich. So Patrick Forterre argues that actually the way a cell responds to its environment and the way a cell interacts with its environment is actually significantly different between a cell that's been infected and a cell that isn't. So he comes up with this idea of the virocell and the ribocell, whereby the virocell will actually interact with the environment in a very different way to the ribocell, i.e. a cell with ribosomes in it. So he believes it's so important that to get this difference in terms of understanding how global carbon biogeochemistry works, that actually we should essentially model for these two things. So the other thing viruses do is that they are, when they replicate inside a cell, that replication tends to be, it's certainly not a clean process. There's lots of DNA moving around and being packaged up. So it's actually relatively common for viral DNA and, for the virus to essentially capture some genes from its host and then put those into the capsid. And then when that virus breaks out and goes on and infects another cell, it can then essentially take those genes from one host and put them into the cell of another. So they are a major shuttle for horizontal gene transfer among the taxa that they infect. But it also goes slightly deeper than that, in the sense, again, this Patrick Forterre put in his paper, because of the messiness of viral replication, it's actually relatively, well, I'm not sure they know the rates, but it's perfectly conceivable to understand the idea that actually when these viruses are replicating, domains from one protein could be mixed with domains of another and actually therefore create new function during the viral infection. So he calls viruses the cradles of new genetic information. And that's kind of, I guess we see evidence for that when we look in viral metagenomes and the number of novel proteins that we identify there that we don't really know what they do. They tend to be extraordinarily high numbers of proteins associated with the virus that we don't really understand what they're doing at any given point. So it might be actually that there's just such high novelty that they're not really hitting anything else in the database. So hopefully now I've convinced you why studying viruses are important. So why are we ignoring them then? Well, I would say the absence is due to largely technical limitations. Firstly, culturing of a virus requires a host, and it used to be the case that the only viruses we really understood anything about were the viruses that we had a host for. So typically, there's an awful lot of viral information out there for your E. coli's, your Pseudomonas, your Sherwinellas, your Vibrios, these kind of things, all the stuff that happily grow on a plate and that you can do plaque assays on. Now, obviously, as anyone who's worked, alongside Cameron and Paul Corrine and these kind of people who understands the idea of the great plate count anomaly, and actually there's an awful lot of stuff out there that is currently resistant to culturing, and there's a lot of work being done now and actually to try and improve that. So until we actually get a host into culture and growing into exponential phase that we can understand it and visualize, it's very difficult to identify viruses from them. So that's probably the primary limitation fact for why we don't really know much about the viral world. There's also no universal marker gene associated with viruses. So I would argue that the explosion in microbial understanding in terms of microbial community composition was largely down to the work by Norman Pace and early pioneers actually looking at, and Carl Woese, looking at using the 16S marker gene as a kind of taxonomic marker for the communities. You can use a single universal gene, amplify it up, sequence those amplicons, and then start to understand what the composition of those communities are based off the amplicons that you see there. Whereas we can't do that with viruses. There are some marker genes that work across relatively narrow ranges of viruses. So I think Meyer's work initially found a phage gene that you can use for some of this work, and then there's some work recently been done on a T4 myoviridae set of primers that target the G23 major capsid protein that pulls out T4 like myoviridae primers. But you're missing whenever you choose to do the amplicon sequencing of these, you're choosing to ignore quite a lot of the viral community. So how do we get around that? This is what we tend to do. So this is metagenomics we tend to use with iron chloride man, and I'll explain why in a minute. So we use iron chloride to isolate our viruses. So this is the guy who's going to come to our rescue in terms of trying to understand viral metagenomics. And in the future, he'll have this hand-mounted MinION sequencer in order to actually sequence these things on the fly, which I'll get to in a minute. So how do we do this? We typically go out into the marine environment. I work. This is our boat out in the Sargasso Sea, just off the coast of Bermuda. And we take around 20 to 40 liters of seawater, and then we pass it through a 0.2 micron filter, essentially to remove the bacterial fraction and hope that the viruses will go through those membranes. We then add iron chloride to that filtrate. And essentially what happens is the iron chloride starts to precipitate, and it precipitates around the viral particles. It nucleates around those viral particles. And then what you can do is actually pump your precipitate out and capture it on 1 micron filters, as you see here. So this spreading of brown gunge in the middle of our filter is actually viruses encoded with iron being stuck onto the filter. And then you very, very carefully peel that filter off of your filtering rig. This is my PhD student Joe, who did much of the work and kind of figured out how to do all this, at least for the MinION sequencing, from borrowing from Matt Sullivan's group. And yeah, you filter it off and you basically try and resuspend that viral particles. Then what you do is you go through various cleanup kits, and what you end up with in the end is your 40 liters of seawater has been reduced to a couple of 100 microliters of kind of brown, cruddy, iron-filled liquid, which you then hand to your sequencing center, and they look at you in disgust as you ask them to actually come out with some DNA from it. So quite a lot of the work we had to do was actually taking something that's this kind of color here and actually turning it into something that would sequence on our platform. So that's essentially what we've been doing. And the largest survey of this globally, I would say, is the Tara Oceans. Now, hopefully, you guys have come across some of the Tara Oceans work before, but it's a European project to essentially, I guess, do what Craig Venter tried to do, but do it properly when they did their global ocean survey, and actually go around all the different parts of the ocean rather than the warm, sunny bits, and actually sample the viral communities and the microbial communities and the algal communities at each of these different points. So every number on here represents a station, and there are various different years where they've been sampled. So for each one of these, there is a virome, I believe, and a metagenome from the cellular fraction for different depths. And then Jem Brum and Simon Roux spent some time actually analyzing this kind of data and started to really figure out what the ecology and the ecogenomics of the viral populations were in the open ocean. And hopefully you can see from this that actually looking at the viral populations, they've done a pretty good job of sampling all the different types of viral populations that you get in the surface water. We're now at the point where that rarefaction curve is actually starting to top out here. So, you know, we don't tend to, when we're sampling, we tend not to find highly novel viral groups anymore in the population level, at least. And then in the mesopelagic, you'll see we're actually not doing a great job of kind of capturing the viral diversity here. It's still going up, so we need to do a lot more sampling in the mesopelagic. And then in the bathypelagic, we're starting to tail off. And that's simply, I think, because there's just far fewer viruses in the bathypelagic. There's far fewer cells down there. Everything grows a lot slower. So they probably, in my opinion, have slightly different infection kinetics than they do in the surface water in the mesopelagic. But yeah, it's a great paper. I highly recommend you go off and check that out, because it really kind of shows where we've come using metagenomics for understanding viral ecology. Okay, there are some limitations, however. We found recently, or Simon Roux found recently, and it was backed up by Manuel Martinez Garcia's group, that actually some viruses tend to fall to bits when you assemble them. Now, this is quite a complex graph from this benchmarking VirIOmics paper. But the bit I want you to concentrate on is primarily this bit here, so over here. And what you can see, hopefully, is if you have a part of your population of viruses is actually relative, so it's not dominated by any specific strain, it's actually got quite a broad diversity of viruses here. If you start to look at an average nucleotide identity of around sort of 95 to 100%, actually the viruses start to fall to bits. And hopefully you can see here that as you increase, almost irrespective of which sampler or which assembly you're using, you can see here that actually we're starting to recover lower numbers of those genomes from the metagenomes. Now that might be a problem, and I think it's a problem because, this is when it was backed up by Manuel Martinez Garcia, when they isolated no, they didn't isolate, sorry, it was a single viral genome called VSAG37F, which they went on to, it found it was incredibly abundant in the oceans, and they claimed it's one of the most highly abundant viruses in the oceans. And what they actually showed is if you artificially increase the microdiversity of this particular thing, then you can hopefully start seeing here it starts to fall to bits. And you can see actually these are genomes here of the particular types of viruses, and you can see some of them are just falling apart. So there's something going on in the assemblies with these that actually is stopping the recovery of these particular genomes. So just the message to take away here is that actually high abundance and high levels of microdiversity is bad. It's bad for metagenomics. It's bad for the recovery of these kind of viruses from the marine environment. The problem comes that actually one of the viruses that suffers most from this is the pelagiphages. So for those of you who are unaware, the pelagiphages infect a group that is colloquially known as SAR11, or historically known as SAR11, but now known as Pelagibacter arce. Now, these are a group of organisms that are hugely dominant in global oceans. They tend to comprise up to 50% of microbial populations. And for a long period of time, people actually wondered why, or hypothesized why it was that these particular organisms could be so abundant if kill the winner was working, and so on and so forth. And one thing they postulated is actually that they were largely immune to viral predation. People had looked through databases. No one had really found a virus that looked like it infected SAR11 or the Pelagibacter arce. So they'd assumed that it had this cryptic escape system. Somehow it grew slowly enough that viruses weren't bothering with it. So I was fortunate enough during my postdoc with Steve Giovannoni, I was working with Yanlin Zhao, and she isolated 4 viruses that infect the Pelagibacter, the coastal strain 1062 and 7211 of the Pelagibacter ubique 1A clade. And what she isolated 4 of them. And then what happened was, once we actually had the isolates and we could sequence their genomes and then use those to track what the abundance of these things were in global metagenomes, we actually found that these are probably some of the most abundant viruses on Earth. And actually, so what had previously been missed was actually hugely abundant, especially this particular one here. This is catchily named HTVC10P that might come up later on in the talk. So this is kind of what was putatively one of the most abundant viruses on Earth. It was around 20–25% of all viruses in the ocean were HTVC10P-like. And E is kind of a cool picture. This is just a picture of a SAR11 about to have a really bad day as all the viruses kind of pop out of it during the infection. So what does this mean for the metagenomics? Well, so for the carbon side of it, we estimate that there's around the same amount of carbon in viruses that infect SAR11 as 45 million blue whales. So I would argue that anyone who isn't particularly interested in the microbiology side of it is interested in blue whales, well, there's probably more carbon, certainly far more carbon in the viruses associated with the bacteria than there are in the precious blue whales. So we should be saving the Pelagibacter arce rather than the blue whales from a carbon perspective. But the SAR11s are incredibly important in terms of their global carbon biogeochemistry, and it's estimated in a recent paper by Steve Giovannoni that up to 5 to 20% of all global primary production is converted back to atmospheric CO2 by this particular clade of organisms. So that includes the terrestrial stuff. So it's not just the stuff in the marine environment. It's actually the entire of global primary production. So they're hugely important. The way it works, for those that don't know, I guess it's Oceanography 101, is essentially the sunlight is captured by the phototrophs. They release dissolved organic carbon. That's then fed off by the heterotrophs, where the Pelagibacter lie. And then essentially the viral shunt is there because the viruses then infect the cells. They break them open. That then releases a whole load of carbon back into the environment. And the viral shunt essentially allows the microbial community to recycle that carbon and stop it going higher up the food chain. So it increases the efficiency, if you will, of remineralization of that carbon back to the atmosphere. Okay, so the problem we have with the pelagiphages when it comes to metagenomics is they're almost completely missed. So if we take this picture here, which is, I guess, a classic 10P-like genome that we have here. Now, we know these genomes have these genomic islands on them. So these are areas where the coverage drops away on those particular islands, and those tend to be associated with, I guess, the arms race between the virus and its host. They typically tend to be associated with things that allow for increased infectivity or that sort of thing. So what we find is if we assemble our short reads from a viral metagenome, and bearing in mind how abundant these viruses are, and then we align those metagenomic contigs back against the 10P-like genome, actually, hopefully what you can see here is that there are huge gaps involved. So there's a large gap over here that simply doesn't ever get resolved by the assembler, and again, some gaps in here around the hypervariable regions. So it's not necessarily just to do with coverage. You can see here where the coverage drops away, but there are some areas where actually the coverage is pretty low, like here, but it's still managing to build a contig across contig across those parts. And in other places, it just kind of isn't, where the coverage is really, really high. So it's also potentially to do with nucleotide diversity. Now, one of the things we find with nucleotide diversity in TEMP-like viruses, it actually tends to be really high across the genome. So these are some of the maximum values we've ever seen in some of the viral contigs. And we think it potentially is this, based off the work of Simon Roux and Manuel Martinez Garcia, that's actually driving the lack of these contigs from actually being able to assemble. And for those of you who don't geek out about assemblies, it's basically because when you do a de Bruijn graph assembly, which we've kind of followed the, I guess it's the encouragement of use of short read technology, simply by the amount of sequencing that you get back, the bang for buck of Illumina, kind of forced us into the route of actually trying to, we could no longer do things by overlapping contigs. We had to start making some rather novel ways of doing it. So the de Bruijn graph was typically used to try and assemble these things. And ideally, what you want from a genome is actually a de Bruijn graph that looks like a single circle or a single line. But when you actually look at the de Bruijn graph of what we get from trying to assemble a TEMP-like genome from these viruses, it actually looks like this here. So it's a big tangled mess. And what's essentially happening is every time might be a contig here, for instance, but then getting to this point, it just simply has no idea which is the correct route to take in order to reassemble that contig. So it ends up being broken in half, broken at this point. So you can imagine how many joins you have here and how fragmented it gets. And then you end up with a whole bunch of stuff here that it just doesn't know where it connected to. So bearing in mind, this is a 29 kilobase pair genome. It's broken up into 2005 scaffolds when we did this assembly, and the longest was 3,527 base pairs long. Now that's a problem because our cutoff for deciding whether something is a viral contig or not, whether we have sufficient information to decide that it's viral, is around 5 kilobase pairs if we're being generous, and 10 kilobase pairs if we're being stringent on our checks. So you can imagine at this point, we're going through all of our big metagenome data. We're then saying, right, we're going to throw away anything that's less than 10 kilobase pairs in length because you can't be sure it's viral. Well, suddenly we're now binning almost all of our pelagiphage data from these environments. So the most abundant viruses on Earth are simply being binned into chucked into the bin of stuff we don't really want to look at because we can't be sure that it's viral. And I would argue that from an ecology point of view, this is problematic. So I guess my group then started looking at that and thinking, well, given that the new sequencing is coming downstream, where we're moving away from short read sequencing and actually now have the capacity for long read sequencing, either through PacBio or doing the new Oxford Nanopore technology stuff, then maybe we can resolve some of these problems using the long read. So the first hypothesis we had is actually, we know that your de Bruijn graph is going to fall apart when you assemble these kind of things, but can you use the long reads to scaffold those contigs in order to build better genomes? Can you bridge some of those gaps with long reads and actually therefore improve the recovery of contigs or scaffolds greater than 10 kilobase pairs? So that was the first hypothesis. But then we also thought, well, in the olden days when people used to use Sanger sequencing and actually do their assemblies that way, overlap layout consensus was the I guess the technique du jour. You used to simply align one sequence against another, and if there was an overlap, you stuck them together. And we kind of moved away from being able to do that as the number of reads increased. But we have relatively few numbers of long reads. So can we actually use that and essentially prevent these parts on the graph that look, or the de Bruijn graph that look like these kind of really knotty areas, and actually properly assemble them with overlap layout consensus using the long reads? So that was the second hypothesis we wanted to test. But then we also thought, well, actually, there's a reason that for many, many years, Craig Venter's trip around the global oceans was actually one of the most used metagenomic data sets that was out there. And the reason is that the reads that they created were Sanger reads. They're about 1,000 base pairs in length. Now, that's just about long enough to capture a gene. So it doesn't necessarily matter if those reads assemble or not. You can still use them to identify what types of genes are out in that particular environment. So with the long reads we're getting now, so PacBio, 10, 20 kilobase pair reads, and then MinION reads, which I'll go into in a minute, can be very, very long indeed. It almost doesn't matter that some of these reads don't assemble. You can still use them in order to generate find out what the genes they're encoding are and understand the order in which those genes appear, so the synteny of those genes in a particular organism. So why isn't everyone doing it, and why is it not all rosy? Well, there are a couple of problems. The first one is, as you're probably aware if you followed the MinION story, the error rate and indels in Oxford nanopore sequencing technology are definitely a challenge for gene calling. They're progressing incredibly well. It used to be, I'd say 2 years ago now, when I did a PhD viva for one of the first papers for MinION, and that was around a 30% error rate. So it's, when there's 4 base pairs or 4 bases, a 30% error rate is not very useful for doing these kind of things. And they're now up to sort of 95% accuracy, 92, 95% accuracy. But the problem they have is that their error model, if you will, is kind of indel rich. And the reason is, I'll go into why in a minute, but it's a bit like the old 454 sequencing for those who know it. It's when you have these homopolymers in there, you don't know how many particular, if you have a run of A's, for instance, you don't know how many A's you actually had going through the pore at any one time. So indels are definitely being introduced into the sequence. And what they tend to do is frame shift. And therefore that means when you're gene calling, suddenly you end up with an awful lot of stop codons in your sequences. Your predicted genes tend to be very, very short. Which causes some significant problems, particularly in viruses, where actually most of what we're looking at most of the time in protein space is actually unknown anyway. So it's kind of difficult to identify what is a genuine short virus protein that we don't understand what it does versus an artifact from having a stop codon introduced through indels in long reads. The other problem we have is actually that typically we take our 20 to 40 liters of seawater and we get around 20 to 100 nanograms of starting material from that volume of water. Now, typically for a good MinION run, you're looking at adding 1 to 2 micrograms of DNA for your starting material. So you can see that we're pretty much out by nearly an order of magnitude for actually the amount of DNA that we need to get into the flow cell in order for it to work. And the reason for that is essentially you need a lot of DNA so they can actually find the pores in the MinION sequencer. And if you don't have any, it just simply burns through the ATP that's on there, and the flow cell dies without really giving you any sequence space. So for those that haven't been following the MinION sequencing, I'll just kind of introduce you. This is the MinION device here that you can see me probably loading water in at this point for a photo. And you can see here that this is the size of it, and it's plugged into a USB onto a computer here that does all the sequence analysis. And essentially in this top device here, in the flow cell, is a membrane. And in that membrane is embedded these pores, this blue bit here. And essentially what's happening is there's a fuel in this particular space, and there's charged ions in this particular space. And you apply a current across this membrane. Now, what happens is the DNA is tethered to a motor protein, that's this particular one here, and then that binds to the pore and starts to feed the DNA through that particular pore. Now, what this is doing is causing steric hindrance inside the pore and therefore blocking the transport of ions from one side to the other. So you can actually identify this in what we call the squiggle space by the interruption of ion flow between them. And what they found is that different bases going through this particular pore actually change the disruption in a different way. So you can use that through some heavy machine learning to actually predict what your base pairs are in this particular point. So you imagine on here in a particular flow cell, you have, I think, 512 pores all running in parallel, and the DNA going through them that way. So a huge volume of sequencing going through. So typically in a 48-hour run these days, people are generating 25, 30 gigabase pairs of data from their reads. Now, hopefully what you can see from this is actually, unlike an Illumina approach, whereby you're essentially having to break up your DNA first into tiny fragments in order to get it to sequence properly on the machine, the length of your DNA here is actually entirely dependent on the integrity of your DNA. So if you have a 500 megabase pair read, for instance, and you manage to successfully extract that without breaking it, and you manage to attach a motor protein to it, at this particular point, there is nothing really stopping that entire read going through and generating a single read for that single strand of DNA. So if you can keep your DNA integrity as high as you can, then you will get very long reads. And the actual record at the moment stands at 2.2 megabases for a single read by Matt Luce's group at the University of Nottingham. And Oxford Nanopore now have this thing where if you get the longest read, you get handed this small cup and you get your name on it, a bit like the Ashes. So for anyone who follows cricket. So yeah, so this is basically why the long reads will actually be better for our kind of stuff than the Illumina sequencing, because you're not breaking it up into tiny fragments and therefore you could potentially capture larger parts of your viral metagenome. However, I touched on briefly, the error rate is around 5 to 8% in indel rich, and that can really screw up gene calling downstream. So it's not all roses at this particular point. Okay, so we knew how to extract DNA from viral metagenomes using iron chloride precipitation. We knew kind of how to sequence on the MinION. So we thought, well, how do we bring these two things together? So we came up with this proposal, this plan to do this initially with the 2D sequencing reads, which are now obsolete from Oxford Nanopore, and then we moved on to the 1D sequencing reads. So 2D is now obsolete, so I won't spend too much time explaining it, but basically that used to read both the template and the complement strand and use it to error correct, whereas the 1D reads only ever read the template strand. So the error rates are slightly higher. Okay, so we take our iron chloride precipitation of viruses, we then extract the DNA from those particular viruses, and then we shear to around 8 to 9 kilobase pairs. Now, the reason we're breaking these things up, it seems kind of counterintuitive in order to, if we want to get full viral genomes coming out of it, but actually it's because we need to amplify this in order to get the right amount of starting material for the Oxford Nanopore MinION protocol. So we shear to 8 to 9 kilobase pairs and essentially perform a linker amplified shotgun library. So we add our PCR adapters onto each of our fragments, and then we PCR through 15 cycles to increase basically the amount of DNA that we've got to put into the flow cell, and then we ligate on our motors, and then we stick it onto our flow cell to sequence for 48 hours. Obviously, the first time you're going to do something like this and kind of change the way you're sequencing, you need to test this, and this is really where our collaboration with Matt Sullivan's group has been instrumental in success. Is that they're able to provide us with a mock viral community whereby they took 6 known viruses and then they basically pulled them all together in the number of virions that they had and then extracted the DNA from it. So we knew exactly what the composition of that community should have been, and the genomes for these viruses were known. So we essentially did a lot of our development on this mock viral community provided by Matt's group. And what we initially found is actually our first worry was that, well, we're adding a PCR step. So we know that when linker amplified shotgun libraries have been produced for short read sequencing, there's been a few papers out now that have shown that actually it doesn't seem to disrupt the relative abundance of it. So there's no bias being added for the, it seems to be amplifying everything relatively equally. So you don't introduce any sort of relative abundance bias. So we thought, well, we're not doing 300 base pair fragments. We're doing 8 to 9 kilobase pair fragments. So are we now starting to introduce some bias? And we're really pleased to find that on our mock viral community that we had, actually, when you sequence these things on the Nextera using Illumina, and then you map how many reads you've got going to each genome, and then you compare it to what you get from the long reads, then your R squared is 0.975. So it's doing a remarkably good job compared to the short read sequence of actually capturing the relative abundance of those particular viruses in there. So we knew at this point we weren't actually introducing, well, we knew 2 things. One, it actually worked, and 2, it wasn't introducing any abundance, any biases in relative abundance through the PCR step. So then once we had that working, that actually, I'd say, was a relatively easy step in that it probably took, I guess, 12 months of Joe's hard work to get the wet work up and working, generating these libraries. And then we thought, well, what the hell do we do with them? None of these tools are actually designed to deal with particularly long reads. They're all designed to use Illumina reads and these sorts of things. So we had to come up with a bioinformatics pipeline that essentially tried to maximize the benefits of both of the sequencing technologies by combining the two. So this is what it looked like eventually after some trial and error. So we have essentially, let's say we take a sample, and one of our test sample that we took was from the Western English Channel, and we sequence it on Illumina, and then we sequence exactly the same sample on the Oxford Nanopore. We then run that through 2 separate assemblies. The first is the standard short read metaspades assembly, and then we run a similar assembly, but combining it with the long reads for hybrid assemblies. So essentially, it's using the nanopore reads to scaffold the contigs from the short read assemblies. So you can imagine now using these 2 approaches, you're actually starting to, some of the contigs you get will be the same in both of them. So you then need to dereplicate these by nucleotide identity. So we typically take a cutoff of 95% identical over 80% of the length of the shortest contig, sorry. And then at that point we say, okay, these are pretty much the same thing. And so we'll take the longest one of those. And more often than not, the hybrid approach here is actually generating the longer contig. There are some times that the shorter one generates the longer contig. We're not entirely sure why. And I did raise this point with the Spades developers, and I never actually got a reply. But about 3 days later, a new version of Spades came out, so I still need to test whether that's actually fixed this problem. But then I also wanted to think, well, okay, let's have a separate route of this. So we can go around this route of taking our long reads, doing our overlap layout consensus in the old Sanger style way of doing it, and then actually using the short reads to error correct the long read assemblies. Now, we knew at this point that this had been done on single cell samples for E. coli and human samples. But actually, I believe this is the first time we tried it on the metagenome. So I was kind of skeptical initially about whether or not this would work. But hopefully I can convince you that it seems to work rather well. And then what we do is you combine all these things together, and then we run VirSort on them from Simon Roux to identify which particular viruses were, which particular contigs were viral, sorry. Those then get passed into a viral contigs database, which you can then use for various viral ecology type tests. So understanding how abundant they are, use Fast Virus Explorer, understanding how they group into genus-like ICTV taxa using vContact2, and then comparing that to other viruses from the GOV, the Global Ocean Virome from Tara, and RefSeq, and various other data sets that we had. Okay, so let's see first. Okay, so the first thing is, does this bit work? Does it work when you go down this route? Does this adding nanopore to your sequencing, does that improve your sequencing of your viral communities? Well, it absolutely does. So there were lots of examples of this, but this is probably my favorite. So using the long read sequencing, so this is a hybrid assembly. So it's standard Spades assembly, where we're actually using the nanopores to scaffold different contigs together. We managed to capture a complete viral genome, 316 kilobase pair myovirus from our Western English Channel sample. And hopefully you can see here in blue that actually when you align these short read sequences against the long read sequences, you can see that in the short read only sequences, when the short read only assemblies, sorry, they're actually breaking the contig at this particular point. So the long read sequencing captures the full viral genome, whereas the short read sequencing only captures it on 2 particular contigs. They're still pretty big, but it's actually failing to successfully span this particular gap here. So the long contigs, the long reads are actually enabling the scaffolding of these 2 things together at this particular point. Okay. The sad thing is that even this approach doesn't really seem to assemble the pelagiphages, particularly Temp with this high abundance, high microdiversity here. So what we can see is even a hybrid de Bruijn graph approach here. So you've got your old short read assembly contigs that we showed previously, and then you align your hybrid assembly contigs to them, and actually you find they're identical, right? There is no benefit gained from using the long reads through Spades to kind of scaffold these things together, which was kind of crap, because it was like, oh, kiss another Nobel Prize goodbye as these things fail to assemble. But then we thought, well, we've actually got a bite of the cherry here. We've got these object layout consensus things that actually should recover from the microdiversity issue. So as long as we can error correct properly, then we should be golden. And what we actually found doing that is that you can see on here in brown, this is the VirION long reads that we had, and those long reads absolutely span the genome of this thing. So you can see through an object layout consensus that you can simply align these reads and therefore reconstruct the genome of the virus. And indeed, that's what we see. So when we do an object layout consensus assembly, whereas before we had like 2007 contigs, I believe, we're actually now getting 1 scaffold here, and it's 28.9 kilobase pairs long. So a full viral genome of a 10p-like virus. Extracted using the long read sequencing technology and then error correction. And going back to the initial point about, well, even if the reads don't assemble, they're still of utility in these kind of processes. You can actually start looking at what's involved in your genomic islands of this. So what we did here is we took our 10p-like genome. This is actually TIG404 here, and then this is it shares a genomic island with same as the isolated 10p strain. And what we actually find is quite interesting. So the internal virion protein here is fairly common in these areas. This is the virus. This is the protein that enables the virus to punch through its host's cell wall and essentially get the virus, the DNA into the cell. But what's more interesting for me, I think, is actually upstream of this is the ribonuclease. Now, the ribonuclease typically tends to be a core gene found on viral genomes because it's used to essentially mediate the host's transcripts. But the fact that it's in a genomic island on the 10p genome suggests that actually somehow the ribonuclease is involved in the arms race between it and its host. Now that's exciting for me because SAR11 is known to not have a lot of cellular regulation. It tends to use riboswitches more than other stuff. So it might well be that actually this virus is using its ribonuclease in order to try and have an arms race, trying to take over the host. And there's this kind of this balance going on between how the host is using its RNA for regulation and how the virus is using it. So we're definitely exploring that in more detail at the moment. Okay, so what about the sequencing error? So we know that these long reads, they introduce indels. So what we did is we essentially said, well, how much short read data do you actually need to error correct these long read assemblies? So we had like originally 30 gigabase pairs of sequence data, primarily because they accidentally run our Illumina at 2x300 on a high seq and end up with a whole ton of short reads. And hopefully what you can see is this dotted line here is roughly the mean size of your average Tara viral database. And you can see that as long as you have that amount of data, so I think it's around 8 gigabase pairs of data, actually you can max out the number of deletions and insertions that you can actually fix in your data. So at that point, you do a pretty good job of fixing it here. So you can see your median coverage is just increasing over these replicates. So we can see these are pseudo randomly subsampled and then used to try and error correct to see what gets fixed using Pylon. And you can see here the total corrected single nucleotide polymorphisms decreases. I think that might be due to the fact that actually you're now subsampling essentially more diverse populations. So therefore you're not correcting these SNPs anymore. They're actually genuine SNPs in the data. But we're still kind of exploring as to why this tails off at this particular point. So then when you translate that into what you're getting out from your predicted proteins, well, hopefully you can see that if you do gene calling on your raw MinION sequences, you end up with these tiny crappy reads here. But actually your overlap layout consensus using Canu does a relatively better job at correcting some of these. These are the Bruin graph assemble contigs. So these are typically your short read sequences, just as we would have done previously. And again, these are typically the types of sizes that you would see. Now, the one to compare this with is the RefSeq cordoviralis genes from, so these tend to be isolated viruses that we have good quality genomes for. And hopefully what you can see here, that if you error correct with the long reads, actually we do a remarkably good job of recovering the similar size of predicted protein length in it. So we're pretty confident that we're doing a good enough job of correcting the areas in order to actually start looking at what these viruses are encoding at that particular point. Okay, so when you start looking at the Western English channel in terms of the ecology, hopefully what this graph shows, if you look at the top 50 most abundant viral contigs in the Western English channel, the vast majority of these are only identified on the long read sequencing. So these are long reads sequenced with overlap layout consensus and then error corrected with Pylon. And every time you see one of these, it means it's outcompeted a short read equivalent of it. So we can see here that this one here is a hybrid assembly, and that seems to be crazily abundant. And again, we think this might be a new pelagiphage we're exploring at the moment. But then you have all these others down here that are all Pylon reads. So these are all long read sequencing reads that have assembled through overlap layout consensus. And then essentially there's nothing on here from GOV or any other. So there's either a lot of novelty in this system, or we're starting to capture stuff using this new technique that hasn't been seen previously. And you can start to get a feel for that when you actually start mapping global ocean virus data from Tara back to our samples. So this is where we are, the Western English channel. And these are our samples here. So you can see, obviously, in our sample, our viruses recruit far more reads than other viruses. So again, we're increasing the abundance there. But you can also see that's the case in other global ocean virome samples. So particularly, I think it's interesting around coastal regions here. We start seeing an increase in the size of the orange, yellow, and lighter orange colors here. So these are all Western English channel viruses that are actually outcompeting the viruses that were taken from that particular site. So they're recruiting far more reads than the viruses isolated from those particular points. So I think we've been missing in the global ocean virome data actually some of these ubiquitous viruses just simply because they've been falling apart. And when you start introducing them, actually they start to recruit a lot more of the reads that are there. Okay, so I'm coming to the end now, but this is kind of where we're heading with this work now. I would say that at the moment, certainly for viral metagenomics and metagenomics in general, we kind of have this weird perverse view where we sequence our data, we assemble it, and then we get very excited when we have these gigabases of contigs all broken up, and we put them through Ambia and we try and solve these things. But I think part of it is kind of where we're going in the future, and I think that's going to be simply using these long read sequencing technology to ideally to capture every genome from an environment on a single read, and then we can simply concentrate on doing the biology on these systems and not worry about the assembly. So if you imagine a million-fold single-cell genomics experiment whereby we can actually just simply look at the population diversity of these things, that's ultimately where we're heading. And we've been trying to work towards that by trying to refine our techniques to remove the necessity to shear these DNA into fragments, and then B, to remove the PCR step. So we've now got to the point where we can actually take our mock viral community, sequence it on the MinION, and capture near complete reads from our viruses. So you can see here, hopefully, these numbers here are the length of the genomes associated on the left-hand side. And hopefully it's obvious there's a big peak around this dashed line. Now, this dashed line is 100% of the genome. So bearing in mind, you've got some indels being introduced into the sequence space, but hopefully you can see we're capturing an awful lot of reads that are the full length of that viral genome at this particular point. And that's less so when you have the big viruses, but this is one I'm kind of proud of, is 128 kilobase pair read that maps about 94% accuracy to this particular myovirus here. So we're getting better at capturing full-length viral genomes on single reads. Okay, so just to conclude, long reads can scaffold the Bruin graft assemblies to improve contact lengths in viruses. We definitely showed that's the case. Long reads assembled by overlap layout consensus. We can deal with the microdiversity using that and therefore start capturing viruses we previously missed. Unassembled reads long enough to provide information of encoded genes and synteny, and we're currently working on how diverse these genomic islands are in some viruses to understand whether it's a protein level or amino acid level diversity that we're seeing there, just using the unassembled reads. We can fix the error rates of MinION sequencing using short read sequencing, and we can capture long read viral genomes, significant increase in novel diversity in global ocean viruses. So again, we've improved on where we were before. And I think single read, single genome metagenomics is coming, and I actually don't think it's that far away, particularly if the progress we're making continues at the pace it is. Okay, just to acknowledge Joe, my PhD student, who does all the heavy lifting here, and then Matt Sullivan and Natalie from Ohio State University, who have been instrumental in getting this work off the floor. Thank you very much.
All right, Ben, thank you very much. Fascinating. Okay, so do you have any questions inside the seminar? Anybody? Feel free to unmute yourself. If I have you muted, you can send me a message. I have a question, Ben. Let's see, can you, you've obviously focused on MinION here, but what about other long read technologies like PacBio? Do you have any experience with that, or how do you think that would fare in this?
Yeah, so we initially started out doing this with PacBio, but the problem we have with PacBio is that in our pipeline that we have here is everything goes through a blue pippin. That's a size selection gel before it goes on the PacBio, and that was largely losing about 98% of our material going onto the PacBio. So we were having to generate huge amounts of material just to go onto the PacBio, which meant more cycles of PCR, which meant more chimeras are being introduced. So realistically, I think PacBio will top out typically around 20 kilobase pairs in length for your reads, whereas the MinION you can go forever, right? As long as you have a contiguous DNA sequence, you can keep sequencing it. So yeah, I don't want to put my neck on the line, but there's probably a reason that PacBio keeps suing MinION. And, I think it's probably to slow them down.
Yeah, well, it's obviously much more expensive too, right? So like the dollar per base pair there is totally different. Okay. Yeah. Other questions for Ben?
I've got a question. Can you hear me? It's Maya.
Yes.
Hey, Maya.
Hey, great seminar. I had a question. So back in the day when people used to do pulse field gels to like look at genome size, I remember all of these precautions that we had to take, like embedding our DNA in like agarose so that we didn't shear it through pipetting. I know so far you've been shearing things to like the 8 to 9 kb for the amplification, but when you get around that step, are there precautions that need to be taken so that you don't shear the DNA and cut off your reads prematurely?
Yes, that's a great question. There's a lot of work being done on this by Joshua Quick out of Nick Lohman's lab in Birmingham. And they recently released a protocol that allows them to do essentially mega base pair above plus reads from E. coli and human beings that we concentrate on. And they definitely use agarose gels and embed the basically do the digest within the gel in order to maintain the integrity of it. So I think that probably works quite well if you have a lot of starting material. I'm less convinced that it will work with a metagenome unless you drained half of Bermuda Atlantic time series into your viral your iron chloride extraction. So yeah. Picking an agarose plug when you can't see anything is going to be a good way of punishing a PhD student who's done something wrong.
Always looking for those. And one more question while I have you. So the reads that were breaking, or the genomes that were breaking that you weren't recovering with the short reads, is there something special about them? Like, do they have more microdiversity than the other genomes, or are they just more abundant than the others, or does it seem to be kind of random?
It seems to be a combination of both of those things. So if you are highly microdiverse and highly abundant, you tend to really suffer from the recovery from a de Bruijn graph, just simply because it can't figure out how to get around that graph. If one of those things is true, they actually tend to recover reasonably well. It's just unfortunate that some of the most abundant viruses in the ocean suffer from that problem. So there's definitely, I mean, they're special in that sense. They're not particularly special in terms of GC content or anything else, but they're definitely special in the sense they fall into this crappy valley whereby they can't be recovered using those kind of assembly approaches.
Cool, thanks.
I had a question for you about sort of trying to push the lower input limit for putting on the MinION. What, I mean, beyond amplification, what are the things that you think are exciting for doing that? I in particular, I had a little bit of time where we were playing with the rapid kits, the transposase-based kits, and kind of didn't have a lot of luck with those at one point because it sounded like there was something going on with Oxford's production of them. Have you used them recently? What do you think?
So again, we had exactly similar problems with the rapid kits. Everything we touch with a rapid kit just killed a flow cell within 15 minutes of it being on. So we kind of abandoned that early on. So we initially started doing this whereby we were trying to optimize or minimize the amount of DNA that we were adding to our flow cells. And what we learned very quickly on is that actually trying to optimize a MinION protocol is a fool's game, because the minute you think you've achieved it, they simply change the chemistry or change the beads or change the protocol, and suddenly you're back to square one again. So what we're doing now with those plots I showed where we're actually capturing the full reads is we just simply add a whole load of carrier DNA to it. So we keep the total amount the same, but we add carrier and then extract it bioinformatically afterwards, and therefore MinION can do whatever they want with their chemistry, and we're not left holding an unpublishable paper because suddenly nothing is where it used to be anymore.
Yeah.
So.
Cool. Okay. Thanks.
Sweet. Are there any other questions right now? Ben, if somebody wants to ask you a question years from now in the future and they're watching it, but now it's like 2019 when they get to the end of this video, how should they get in touch with you?
Well, Brexit would have happened by then, so we won't have electricity. But
so they need to come find you in the woods.
Yeah, so send by pigeon. But if not, then email at my Exeter address, b.temperton@exeter.ac.uk is a good place, or hit me up on Twitter. It'll be fine. But
all right. Hopefully Twitter will still be there. Okay. Well, if there's no more questions, thank you again, Ben. This was wonderful, and look forward to seeing the many new viral discoveries that you'll be generating with this. This is exciting.
Cool. Thanks for the opportunity to talk. It's great.
Okay. Cheers.
Cheers.